to introduce Amy Miller. And Amy completed her Bachelor's of Science at Ohio State University in 2011 and her Master's of Science in Forest Biology in 2014 at Purdue. And then from 2014 to 2017, she was a STEM and Outdoor Education Coordinator in Carroll County here in Ohio. And in 2018, she worked on the U.S. Brave Niagara. And I encourage you to ask her about that adventure on the tall ship. And then she decided to return to graduate school in 2019, right? Yeah.
I think we just talked about it in 2018.
Yeah, that's right. And the reason why she decided to return is because I was unable to diagnose a sample that her father, Greg, brought in on chestnut. He knew more about it than I did. And, you know, I just kind of said, well, this is a job for a graduate student to do. And a few months later, Amy joined my program. And we're really happy to have her here teaching me, because I knew nothing about chestnuts when she started. And it's been really exciting to learn about chestnut production, but also work with her to really understand this new disease of chestnut. So Amy published a first report of chestnut anthracnose in the United States, and she's currently working on a feature article for plant disease that we hope will come out this fall. She also has published in trade magazines like The Nutshell, and she's received grants to support her research, which is always helpful. She received a graduate student seeds grant, and she also received Paul and Edna Warner endowment grant. And then in 2021, she was the recipient of the Fruit and Vegetable 40 under 40 class of 2021. So that was an award for her work with the industry and academia and contributing to chestnut production and chestnut health across the United States. So after her PhD, she's planning on returning to Carroll County, and she'll be managing the cooperative, Route 9 Cooperative, and also conducting research on her farm to promote plant health and crop production, and hopefully working a little bit with me still to get some of my apple research continued, since people have left me.
But thanks for coming back. Just right in. Yeah, I knew I should have done it.
Yes, if you haven't had a chance to go to the farm, Amy and Greg are wonderful hosts, and I know that they would love to have you out there, especially at harvest time to help pick. And today, Amy is going to be presenting the culmination of her research towards earning her PhD. And the title of her dissertation is Biology and Characterization of Chestnut Anthracnose in Culinary Chestnut. Take it away, Amy.
Thanks. Thanks very much, Melanie. It's been really fun to work in this disease system for the past 4 years, and it's pretty exciting because this is a disease that was not really previously characterized in the literature. It was described, the disease symptoms were described, but there was not much known about it. And so everything that we've been doing in the last 4 years is pretty foundational. We relied a lot on observation to establish some very baseline understanding of the pathogen and the disease cycle. And so we'll kind of walk you through the various steps that we went through to understand this disease and this pathogen a little better. And basically, you know, in the end, now that we've had 4 years of work on this, I feel like now we're ready to just begin the actual research to understand, you know, to really do some future testing. So as Melanie mentioned, maybe we can continue working on this in the future here and there as I'm at the farm. But yes, let's go ahead and get started. Yeah, as I mentioned before, folks on Zoom, if you have any technical issues or questions or whatever, feel free to put those in the chat, and we'll help address those. My lovely Gloria and or I will help with that. So feel free to put stuff in the chat if you need to. Okay. So as you can see here along the top of the screen, these are chestnuts that are symptomatic with chestnut anthracnose. So this is a disease that we notice at harvest time in the fall. Basically, these chestnuts, they normally have this nice kind of chestnut brown color, but we notice these black tips in the nuts. And actually, it's kind of a dry lesion as well. If you kind of press on the nut with your fingertip, they're sort of soft there. And you can see we see disease, everything from this little tiny black tip all the way to almost the entire kernel covered with a disease lesion. This disease was formerly known as blossom end rot, basically because it was a rot. This is the tip of the chestnut is the blossom end of the nut. And so when it was first characterized in 1958, they were really just looking at the disease of the kernel. It happened on the blossom end of the nut, so they called it blossom end rot. We have since renamed this chestnut anthracnose. Get rid of this. Chestnut anthracnose, because we have found this disease not only affects the chestnut kernel, but also affects other chestnut tissues and is found throughout the chestnut tree, whether it's symptomatic or not. And so we've renamed this. We've changed the name from blossom end rot to chestnut anthracnose, which better describes sort of its necrotic tendencies throughout the chestnut tree. And for some background information, so these on the map here, these are locations of 4 different chestnut orchards. This is our location, Route 9 Cooperative in Carroll County, Ohio. At each of these locations, there are maybe 50 to 200 acres of chestnut trees planted. And all of these growers are either members of Route 9 Co-op or friends, or people that are in close communication. And starting in the early 2010s, all of these growers in these fairly spread out area noticed a big increase in this disease that they then called blossom end rot. And this was a disease they had seen previously in the orchards, but it was always less than 2%. It was considered a minor disease. It was something that was noticed, but was never considered a major issue. But starting in the early 2010s, around 2011, 2012, growers were seeing disease incidence above 10%. 10% or more of the crop was affected. And so these people all started talking to each other, and they realized that sort of the patterns of disease fluctuation, years when disease was bad and years when it was not so bad, were actually kind of the same across this rather large geographic area, which was kind of interesting. And so these growers wondered, okay, why is this disease that we've seen before such a problem now? And also, how can we predict disease? How can we know if we're going to have a bad disease year or not? These growers also tried some fungicide applications, things that are used in other, fungicides used for apple scab, for example, or other sort of common orchard diseases. And they didn't really see any effects on disease management from their sprays. And so this was basically a mystery. This kind of culminated in 2018 when we at Route 9 Cooperative, we lost half of our crop to this disease, which was pretty significant. And it not only do you lose half the crop, but you can't see the disease until harvest time because the nuts are concealed in the burrs until it's actually harvest time. And so you don't know how much disease you have until you're already harvesting. And basically you're bringing in all of the nuts from the field. And so you're harvesting half of a crop that you can't sell. So it's kind of like, it's like a 3 times bad thing, And so that was where Melanie and I got involved. Well, after we had that horrible disease incidence here in 2018, we said, okay, we got to figure out what's going on here. Especially because I was moving back to the family farm, wanted to be a chestnut grower. Said, okay, well, I can't grow chestnuts if we're going to lose half the crop every year. This is ridiculous. So this was partially an attempt to help us figure out how to continue to grow chestnuts in our locations. So if we're going to try to figure out what's going on with a new disease and a new pathogen, we'll start with a fundamental paradigm of plant pathology, and that is the disease triangle. So in order to have disease, you need a susceptible host, a virulent pathogen, and an environment conducive to creating the disease conditions. So in this case, we identified the disease, or we knew we had this disease. Basically, this is a chestnut cut open, cut in half. And this top brown part is the chestnut anthracnose lesion. And then this yellow down here is the healthy tissue. But you can see about half of that kernel has a lesion. So this is what we were seeing from 2011-ish all the way through 2018, 2019. Up here, this is an example of healthy chestnuts. So our host crop in this case are Castanea species. Castanea is the genus of chestnut. And we're working mostly with Castanea mollissima, which is the Chinese chestnut that's primarily used for culinary nut production in the eastern U.S. because it's resistant to many diseases that other species such as American chestnut get, like chestnut blight. So we typically grow Chinese chestnut because it's resistant to most diseases, but this disease seemed to particularly be hard on the Chinese chestnuts. So we knew our host, our Castanea species, and we knew what our disease looked like. So then that led us to the question, okay, what is the pathogen and what is the environment that's conducive to disease? The pathogen had been previously identified as a fungus in the genus Colototricum. And Colototricum fungi are really interesting because they are found around the world and they are pathogens of most major food crops around the world. There's some sort of Colototricum that will infect just about anything. And the tricky part about Colototricum, you can see here, here's a image of the fungus growing on a plate of PDA in culture. This is the top of the isolate. This is the bottom of the isolate. And then these are the spores, the conidia that kind of splash around in the environment and cause infection. Many species of Colototricum have very similar morphology. So you can't just tell by looking at these images with microscopy whether what species this is. So this had been identified putatively as Colototricum sporoides, which is we now use it as kind of an umbrella term for this type of morphology of Colototricum, but we needed molecular means to determine what species it actually was. That had not been done previously. And then we really didn't know much about the conducive environment to disease. So we wanted to characterize the pathogen and then also explore environmental factors that may be contributing to disease. So the first step was characterization of chestnut anthracnose. So for this, we wanted to collect and identify pathogens from symptomatic chestnut tissues, and then also demonstrate Koch's postulates, and at the same time do some pathogenicity tests to try to understand how infection occurs. We wanted to examine disease characteristics and storage. This turned out to be a really interesting component of the research because many Colototricum pathogens that affect other fruit and nut crops are known as storage pathogens. So even if you don't see them in the field at harvest time, a lot of times they can cause post-harvest storage rots. So we were really curious. We were seeing these disease symptoms at harvest time, but we were curious to know if this would expand or change in storage. And then, a big question for the growers was host cultivar susceptibility. So are there cultivars or species of chestnuts that are more susceptible or more resistant than others? So this is, host resistance is one of the most fundamental ways to manage disease. If we can just plant resistant hosts, then that saves so much hassle, in other types of disease management efforts. So we were really curious about host cultivar susceptibility. We examined host cultivar susceptibility through disease incidence and severity. Disease incidence is basically the percentage of the crop or the percentage of each cultivar with disease nuts. So we would collect anywhere from 200 to 1,000 nuts from each cultivar each year and just count the number of nuts that had anthracnose symptoms like these. And then incidence is a proportion of the total that had disease. And then after that, we would look at severity. So we took all of the nuts with anthracnose, cut them open sort of along the center of the disease lesion, as you can see in this photo here. And we would measure the length of the kernel, we would measure the length of the lesion, and then express severity as a percent of the kernel that was infected. And then we would average that over all of the infected kernels per cultivar. So we had incidence and severity data for each cultivar for each year from 2019 through 2022. And we chose cultivars of interest, ones that are of economic interest and that are well known to growers that many people have planted, and some newer ones that we've been developing just to get a range of see what the range of disease incidence was among the cultivars. Our site, so most of the samples were taken and processed at Route 9 Cooperative. So that's our processing facility in Carroll County, Ohio. These orchards, these were the sites where these cultivars were planted. And so we harvested nuts from specific cultivars in specific sites. And it was really interesting. We wanted, if we had the same cultivar across multiple sites, we wanted to examine that as well to see if there were differences, not just among cultivars, but also among sites. There we go. I'm gonna make this go away.
Let me go to more. And then the hide floating medium controls.
There you go. Thank you. So as I mentioned, in order to identify the pathogen, we needed to collect disease tissues. So we would collect symptomatic chestnuts. We also found the same pathogen causing disease on chestnut twigs. So we collected samples from diseased twigs. We would plate those bits of tissue, especially we would kind of make cuts of tissue right along the margin of necrosis and culture that on potato dextrose agar. We found that to be a really good medium for growing Colototricum, which is convenient because it's readily available. And then we would isolate all of the fungi that grew from there. And as you can see in this example, these are actually pieces of diseased chestnut tissue that are plated. And there is just one type of fungus growing from there. So it was pretty easy to identify the pathogen from the diseased chestnut kernels because there was really nothing else growing from those chestnuts other than our pathogen of interest. We identified, so then we would grow these in pure culture, extract DNA, and we sequenced 4 different loci to identify the species. So this ITS, GAPDH, beta-tubulin, and APMAT. And these are all loci that have been identified in the literature as suitable for identifying Colototricum species. Not only do Colototricum species look alike microscopically, they also can have a lot of overlap genetically among certain common regions, such as the ITS region. So we actually needed to sequence at least 4 loci to be confident that we had our species identified correctly. So we used those 4 loci, and from all that, we determined that the causal agent of the anthracnose in the chestnut kernels and the chestnut twigs was a species called Colototricum henninense, which is really only described in the literature as affecting tea trees in China, and I think Japanese thistle. And I think there's one report of it in China on Chinese chestnut leaves, but never on nuts. So this is a species that's not very well known. It has been identified in the US as causing bitter rot on apple, but there are many different Colototricum species that cause bitter rot. Colototricum henninense does not show up very often as a causal agent of bitter rot, only occasionally. So this is a species that is not very well studied, and we don't really know much about it other than a few hosts, and we don't know much about its ecology. Seems to be very specific to chestnut though, because we found it all over the chestnuts. So as I said, we were surveying different symptomatic chestnut tissues, and we identified Colototricum henninense associated with disease lesions in the kernels that, this is the outside of the shell, the inside of the kernel. We found it associated with disease on twigs. These are young, maybe 1-year-old twigs, maybe about the diameter of my finger, and the disease lesions are maybe 1 centimeter or so long. So we found them on twigs. Sometimes the lesion would actually, it seemed like it would girdle and kill the whole twig distally from where the disease lesion was. But sometimes, like in this case, you can see that this disease lesion is being walled off. So this plant is able to kind of contain and recover from the lesion. So that probably depends on the health of the tree initially. If it's stressed in other ways, it might succumb to this disease. But if it is otherwise healthy, it might be able to wall off the pathogen and just keep this canker relatively small. But even if it's a relatively small canker, you can see here are some fruiting bodies starting to erupt from this canker. So even if this is not necessarily killing the twig, this could provide spores and provide inoculum to spread disease further throughout the orchard. We were unable to find specific conditions which cause sporulation from cankers in twigs. But given that we saw fruiting bodies, we presume that there are some sort of conditions out there which could cause these to sporulate and potentially spread the disease around the orchard. The other interesting thing, this nut here, so these kind of circular purplish orange lesions, these occur just in the shell, or they seem to start in the shell. And they may or may not penetrate all the way through to the kernel. So these kernel infections here, they appear to start on the inside of the kernel and spread outward. These shell lesions appear to start on the outside of the shell and spread inward. So we've got at least 2 different infection pathways on the kernel and the nut itself. And either way, if you're processing chestnuts, if you see these spots or these spots on the shell, you want to call those out, and you don't want to sell those to customers because you don't know if they're bad on the inside or not. So either way, this represents an economic loss to growers, whichever infection pathway. The other thing we found was looking at these are chestnut burrs. So this is a normal healthy burr around harvest time in late September. These are smaller burrs. You can see they're kind of brown. They have these necrotic areas. They drop early. So they drop maybe a week to a month before normal harvest time. And they have these necrotic lesions. And so we isolated fungi from those necrotic lesions on the burrs. We also isolated Calotetricum henninens from these necrotic burrs, but we isolated other fungi as well. And we were unable to confirm Calotetricum henninens as the main causal agent of the burr necrosis, but it does seem to be related to this burr necrosis as well. So as I mentioned, it's kind of throughout throughout all the different chestnut tissues. And so pathogenicity tests, we wanted to demonstrate Koch's postulates to confirm that Calotetricum henninens was indeed causing disease. And we also wanted to explore mechanisms of infection into the twigs and into the nuts. So we tested the burrs. So these burrs were collected. They were not quite ripe yet. They were maybe a week or 2 weeks before harvest. So they were almost ripe, but not quite. We collected them, brought them into the lab, made an inoculum solution of the pathogen, and just injected them into the nut through the burr, into that kind of developing nut. Held everything in a moist chamber, and then examined disease development 3 weeks later. We also tried non-invasive inoculation techniques where we actually just spritzed, there's kind of misted inoculum over the tops of the burrs. There's an area called the tuft where kind of the little pointy part of the nut, the tuft sticks out, and it's visible from the outside of the burr. So we wanted to see if we could inoculate non-invasively through the tuft area. And as you can see here, wherever we wounded and inoculated, you see this nice like disease lesion developing. But wherever we tried the non-invasive inoculations, we did not see any disease development. So in both the nuts and the twigs, you can see there's a little hole where we injected the inoculum. Here we actually made a hole and put some of the mycelia from the auger onto the wound. Everywhere we wounded, we were able to cause infection, but any non-invasive technique did not cause infection. So that was really informative. It does appear that wounds are possibly really important to the disease ecology. We looked at disease characteristics and storage. So as I mentioned, many Calotetricum pathogens cause disease that expands or initiates in storage. We wanted to look at that. So basically, this is an example of a healthy chestnut. So this was not diseased at harvest time. Chestnuts like this, healthy chestnuts, were kept in storage both for 30 days and for 60 days, and we cut them open and examined them at that time. Anything that was healthy going into the cooler looked pretty much the same after 30 to 60 days in storage. So we did not see any new initiation of disease on nuts that were asymptomatic at harvest time. We also wanted to look at, I'll say this too. So these down here, these black tips, that's from anthracnose. And then these kind of the white mycelia, and there's some different colors, some pinks and purples and that sort of thing. Those are secondary molds. So after about 60 days or more in storage, any nut with anthracnose the lesion starts to be overgrown by other spoilage fungi. And we were able to culture some different types of fungi, Fusarium and Neurospora, and some other things from those lesions. And so these do seem to be more susceptible to post-harvest storage molds than the healthy, non-infected nuts. We wanted to look at storage under different temperature conditions. So these were diseased nuts with anthracnose that we put in storage at 3 different temperatures. These were kept in cold storage at 4 degrees Celsius. These were kept at room temperature, about 18 degrees Celsius. And these were kept in an incubator at 26, which that temperature sort of represents like a warm autumn day, sort of like the ambient warm conditions in autumn. So we wanted to see what happens to these diseased nuts in storage at different temperatures. Interestingly, these ones that were in cold storage, after 30 days, they look very similar to the ones that just went into storage. We did not see any progression of the disease lesion beyond the original lesion when it went into storage. So we don't see any evidence in cold storage that the disease progresses beyond what we originally saw. In both warmer temperature conditions, in the room temperature condition, you can still see this is like the lesion of anthracnose, but then this is kind of a different color, and these kernels, it's not obvious from the photo, but the texture really changes. And then in this warm condition, we see this like total deterioration of the kernel, very different colors, very different textures. when we culture fungi from yeast, from nuts and yeast conditions, we find all kinds of other spoilage fungi. So it appears from our storage research that Colotetricum, the anthracnose does not initiate in storage, and actually nuts with anthracnose that are in storage seem more susceptible to other post-harvest diseases, and we don't see the anthracnose itself expanding beyond the original disease lesions. It's very unusual for Colotetricum species, so yeah, that was kind of eye-opening. This is the part of the presentation that's usually of most interest to growers. So this is our study of post-cultivar susceptibility. So we wanted to know if there were cultivars that were more or less susceptible to the disease. These data, so each bar represents the percent disease incidence at harvest. This is averaged over 4 years of looking at, analyzing disease incidence at harvest. First year we have, this is a cultivar called peach. It's actually a chestnut, but it's called peach. This is peach in 3 different locations. So we see peach with high disease incidence in 2 locations. It's a bit lower at this location. This next cultivar is called Ching. We have it in 2 locations, and we see almost no disease after 4 years in Ching. Get in, same 2 locations. We see some variability among locations. And then these other cultivars we just had in 1 location. It's interesting that we did see significant differences in disease incidence among cultivars, and they were fairly consistent throughout the years. Peach obviously is one of the more susceptible cultivars, and we saw it consistently more susceptible over the years. Ching is one of the consistently least susceptible cultivars, and we saw almost no disease in Ching over 4 years. And another interesting thing, these over here, you see this is the PQ seedlings. These are seedlings that are full sibling crosses of the peach cultivar and the Ching cultivar. And we just pooled the nuts from all of those offspring together because they had just come into production, so each tree didn't have a lot of production itself. So we pooled them together, and we see moderate disease susceptibility in those cultivars. So it'll be really interesting in the future to continue to monitor that population and look at individual trees, these incidents in individual trees, to see if we see any clear differences in disease among those offspring of a highly susceptible cultivar and an apparent resistant cultivar. Another thing I want to point out here, this is the anthracnose severity. These are the data from that. So as I mentioned, we looked at the percent of each kernel that was infected. Interestingly, although we saw significant differences in disease incidence among cultivars, we didn't see differences in severity. So in any given year, the anthracnose lesions are more or less the same size among the different cultivars. It was interesting, I'm not really sure what this means, but this is 2019, this is all the cultivars pooled together, 2019, 2020, 2021, and 2022. These 3 years here, mean severity was more or less the same, but 2022 it was significantly lower. And we saw very low disease incidence in 2022 as well. So just overall, in the last year, we saw much less disease, both incidence and severity. So key findings from the characterization, we found that Colotetricum hananex causes chestnut anthracnose, which affects kernels, twigs, shells, possibly burrs. We were only able to induce disease by inoculation through wounds. No, we didn't see disease from non-invasive methods. The anthracnose appears to infect the nut while on the tree and does not initiate or expand in storage. And we did see differences in disease incidence among cultivars, and some variation among the same cultivar between different sites. So that's also interesting and warrants further exploration of the environmental factors involved. Which takes us to the disease cycle. So now we want to know, okay, we know that there's Colotetricum is out there, it's everywhere. Where is it living in the orchard? What are some potential sources of inoculum? So that objective 1, we wanted to survey and identify what species were present throughout which tissues in the orchard. Where is it? We wanted to identify the optimal growth and sporulation conditions for the fungus, so we know what could cause it to produce spores to spread the disease. Then we want to begin to decipher the infection pathway. So once the spores are in the environment, how do they actually get into the nut and cause disease? And then objective 4, can we put it all together into a model? Can we say, okay, we know these are the conditions for sporulation. These are the tissues that are most susceptible. Can we model and predict how bad disease is going to be at any given year based on those parameters? So we surveyed blossoms and developing nuts. So this is it. This shows you, this is an example of in 2019, the sampling points. So these are chestnut flowers. These are female flowers and male flowers. And you can see this is approximately every 2 weeks as those flowers develop into chestnuts. You can see that little flower develops into a burr, the shells grow, and then here in like late August, early September, we begin to see the kernels develop inside the nuts. And then here at harvest time, you can see the fully developed chestnuts. Some of them are not symptomatic, some of them have anthracnose. So we surveyed the developing nuts all the way from flower to ripe fruit. We wanted to find at what point do we start to see anthracnose in the nuts. We expected maybe there was some point in time where it suddenly showed up, and that might tell us, okay, this is the point of infection. Actually, what we found was it was present throughout the tissues, throughout all the times we sampled. So even these flowers, at the early stages before bloom, we found anthracnose in that green tissue before bloom. So that really let us know that this fungus is an endophyte. It's living inside the chestnut tissues. Whether or not it's causing disease, it seems to be always present in the orchard, which that caused us to kind of shift our thinking about how we approach this disease and what we might, how we might imagine managing it and dealing with it in the orchard. We also were curious about chestnut galls. So this is a chestnut gall. These are some purplish ones, some greenish ones. These are structures induced by the Asian chestnut gall wasp, which is an invasive insect that kind of commandeers the vascular tissue of some of these of the chestnut trees, causes this gall formation. This is a cross section of a gall. This is a little wasp larva that lives inside. So the wasp larva lives in here from bud break in mid-May all the way through about early July, right about this time of year is when the adults emerge. And then once the adults emerge, then these galls die, and you have these kind of dried up sort of mummy galls here. So this is a current year's gall, and this is a gall from a previous year. And the reason we were interested in the galls is that the 4 locations that I showed you in the beginning, those different chestnut growers, they all started seeing chestnut anthracnose about the same time as they started seeing chestnut gall wasp. And so we wanted to know if there was any relationship between chestnut gall wasp and chestnut anthracnose. We also saw these lesions. A lot of galls have these kind of like dark fungal lesions. And it was hypothesized that this lesions were caused by a species of Calototricum. So we thought, oh, wouldn't that be crazy if this thing that's the biological control of the gall wasp is also hurting the chestnut crop? Like, ooh, what do we do with that? So this is another example. This is a lesion, a sporulating lesion on a gall. We looked at it under the microscope. Sure enough, those look like Calototricum spores. We did molecular analyses, and interestingly, we found it was a different species of Calototricum that was causing the lesions on the galls than it was causing infection in the nuts. This was Calototricum fiorini, which is kind of known as a fruit pathogen throughout several different crops. So we have these 2 different Calototricum species causing disease in different types of chestnut tissue. And this is an example of, as we're collecting tissue and cultivating it and isolating fungi, these kind of grayish, white, fluffy looking cultures are the Calototricum henenense. That's our major pathogen. These kind of pinkish ones are Calototricum fiorini. That's the pathogen of the chestnut galls. And then we had some lookalikes, like these kind of gray, fluffy ones look similar to Calototricum. That's actually Alternaria alternata. Once they sporulate, you can tell really easily, but before they sporulate, they look kind of similar. So we ended up culturing a lot of Alternaria as well, just to verify whether or not it was Calototricum. These are the tops of the isolates. These are the bottoms of the isolates. So you get an idea of what we were seeing as we were culturing these fungi. We also wanted to know about sporulation. So what causes these to sporulate in culture and also in nature. This is a culture plate. You can see all these little kind of beads of water. It looks like those are the sporodokia that are producing the spores. So this isolating culture is just covered in spores, and we would use these put some sterile water on there and scrape to make our inoculum. It was kind of interesting. Based on the literature, I assumed that these species might need a light and dark regime to sporulate. I thought they might need higher temperatures. I was spending a lot of time with the light regime, trying to get the temperature right, with these certain isolate, the culture plates. Meanwhile, I had a plastic Ziploc baggie full of isolates and culture. This kind of in like a cool corner in the dark that I was kind of ignoring. Fussing around, couldn't get my cultures to sporulate, walked over, opened up that bag. When they're all covered in spores. All the plates look like this. So, okay, well, that caused us to start to look more closely at the conditions leading to sporulation. We looked at culture plates under different temperatures, in the dark and under high humidity conditions. And we actually found that 20 to 25 degrees Celsius was the optimal sporulation temperature for this fungus. Which was different than our original hypothesis. We thought it would need warmer temperatures. So anyway, we found this is a very temperate fungus, and it kind of likes those cooler, 20 degrees Celsius, about 68 degrees Fahrenheit, like very comfortable temperatures. In nature, so when we were looking at nuts at harvest time, for 2 years, 2019 and 2020, we didn't see any sporulation on the nuts, on the lesions at harvest time. Oh, that's kind of odd. How do these things, how does this move around in nature if it's not sporulating? Well, in 2021 and 2022, we began to see these you see these like pink slimy spots all over the disease lesions. There's some pink slimy spots here, pink slimy spots there. That's actually sporulation from the nut lesions. And we began to see those in 2021 and 2022 and had seen them previously. We thought, okay, well, what can we find any differences in conditions among those different years? Analyzed a lot of weather data, ended up looking at leaf wetness, which we have sensors in the orchard which can monitor leaf wetness. These are used to predict disease in other fruit crops, such as apple. So this was kind of interesting. But we're looking at here, so the orange line is 2020, this blue line is 2019. In 2019 and 2020, we had much more fluctuation in leaf wetness. So basically, there were several days, many days, where the leaf wetness dipped below 10 hours per day during that time period. If we look at the gray line, which is 2021, and the yellow line, which is 2022, those we never see any dips below 10 hours a day of leaf wetness. So we have these sustained periods of free moisture that seem to be associated with sporulation in the nuts. So that was very informative, and that helped us when we were trying to further model the disease cycle. And as I mentioned, so then we were searching for this infection pathway. So we know some conditions which the fungus prefers, and then how does the fungus actually get into the nuts? As I mentioned, we were looking at the fungus coming in through the tuft of the nut and also through the flower at bloom time. This is a common infection pathway for many fruit crops. This graph here, this shows us in one of the orchards, there's a whole block of seedlings, evenly spaced, evenly aged. And I observed that they were in very distinctive phenological groups. So they bloomed at different times. So this group represents the early bloomers. Here's the mid bloomers, and here's the late bloomers. And so we collected nuts from each of those, and we at harvest time, and looked for differences in disease incidence. And that basically, what I was looking for was say, okay, well, if all these are blooming at the same time, and if they all have similar disease incidence, then maybe there's something about that weather condition during that bloom period that would help us know how to predict disease. What we found was, regardless of bloom group, we see this huge range of disease incidence. And here in this late bloom group, we had some that had no disease and some that had 25% disease. So the fact that we have that much variation in disease incidence when they all bloomed at the same time was a strong indicator that conditions at bloom were not predictive of disease at harvest. So we decided to dig a little deeper and try to see if we could model conditions during the growing season, weather conditions that might be associated with bloom time. We looked at, or not bloom time, but disease at harvest. We looked at 4 different phenological windows. The leaf out period, sort of May 10 to June 10, sort of early to mid-May, early to mid-June. The bloom period, which is like mid-June to mid-July. The summer growth period, which is sort of mid-July to late August. And then that fruit fill period, which is late August to late September. And we looked at different types of rain and humidity and temperature, ideal temperature conditions for Colototricum henenense during those periods. And we wanted to see if there were any associations between those conditions and any of those growth developmental periods and disease. And we compared that to disease incidence. We had data from Route 9 Cooperative from 2009 all the way to 2022. And this is disease incidence. You can see as a percent. You can see like it was low in 2009, 2010. It got higher in 2011, 2012, 2013. Kind of dropped a little bit from 2014 through 2016. Came up again in 2017 through 2018, and then dropped again in 2019 through 2022. And then, like, as I mentioned, in the last couple of years, we haven't seen very much disease. So we looked at different weather parameters across those years, across the different developmental windows, and sort of crunched it all together. These were the parameters or the variables that came out as potentially associated with disease. But in the end, we didn't find any strong, compelling evidence that any of these weather factors that we identified were strongly associated with disease. Interestingly, we really didn't find very much relationship between bloom conditions and disease. So we've essentially ruled out the idea that infection happens at bloom time, which leaves us reeling a bit because that was really the most likely scenario based on diseases in other crops. So now that we think that these are not being infected at bloom, we have to kind of reimagine how the infection might be happening. This fruit fill window, that kind of late August to late September, when the nuts are really developing inside the shells, there appeared to be some associations there. And that whole period probably warrants future studies. So we're going to look more closely at fruit fill in the future. But we also think that infections could potentially be happening inside the tree from Colototricum that's already living in the tree, that's going to grow into the kernels through the vascular tissue rather than spores that are coming from the outside. So the key findings from the life cycle studies. So this Colototricum henenense is a ubiquitous endophyte throughout the orchard, and it can live asymptomatically in chestnut tissue. So we find it there whether it's causing disease or not. Colototricum featherini is also found as an endophyte, not associated with chestnut anthracnose, but it is associated with those necrotic gall lesions, which is kind of interesting. Colototricum henenense sporulates in culture at 15 to 25 Celsius, optimally at about 20. And then we observe this heavily sporulation in the field associated with long periods of free moisture and leaf wetness. Inoculation studies and analyses of weather conditions lead us to rule out the bloom time as the main infection period. And then our analyses between weather variables and disease also help support our decision to rule out bloom as the bloom as the main infection court. And this helps us narrow down time windows and conditions to study for future disease modeling. So like I said in the beginning, basically now we can get started. Now we know enough to begin. So what we've, in the end now, after 4 years of looking at this and basically having our hypotheses overturned by field observations and trying to pay attention to what's going on in the field and with the fungus in culture, we determined that this anthracnose disease cycle does not seem similar to other common Colototricum-based diseases such as bitter rot in apple or anthracnose in almond, nor does it seem similar to chestnut brown rot, which is sort of a similar looking infection that actually occurs through the chestnut bloom. Since we saw low disease in recent years, we've had really good growing conditions and really nice weather. We've had very low insect pressure, and we've seen very low anthracnose disease. So that indicates to us that trees that are in very good growing conditions and are very healthy otherwise seem to be more resistant to disease. So that, we're advocating for holistic tree health as a management strategy at this time. The differences in disease incidence indicate that breeding for host resistance is possible. As I mentioned, we had that population of seedlings that are crossed between the least susceptible and most susceptible cultivars. And so it'll be interesting to see how that plays out in the future if we can effectively breed for resistance, because then that would be, from a grower's perspective, plant the resistant cultivars and don't worry about it. That's ideal. we still need to, oh yeah, and exploring those mechanisms of resistance would be a great next step as well, to know, for example, that cultivar chain that we never saw disease. Well, why is that? How is that? How does that compare to peach, right? So that's definitely an avenue for future research. Deciphering the disease cycle, it's still critical to management of the disease on susceptible cultivars. So we, as I mentioned, we hypothesized that infection is actually occurring through the vascular tissue of the tree, and that this Caulototricum species is living asymptomatically as a biotroph, and something is triggering it to switch to necrotrophy. Basically, it's switching from being sort of like a passive occupant in the chestnut tree to being an antagonist. And we don't know what exactly causes that switch. So that's definitely something that could warrant future research. There are several fungal pathogens of various crops that are known to have the switch between biotrophy and necrotrophy. And it's a pretty interesting emerging field of research. And so I think this is a good candidate to throw in with some of those research projects that are looking into that, the switch in lifestyles. And with that, I would like to thank my committee, some are here, some are on Zoom, for their guidance and help throughout this project. Thank the Fruit Pathology Lab for their help over the years with various aspects, folks who are here and folks who have gone before. And big thanks to Route 9 Cooperative. Really, this could not have been done without the support of the co-op and the board members who really supported research in the orchards, and the various employees over the years who have helped with different nut collections and disease incidence ratings and that sort of thing. So big thanks to Route 9 Co-op. As Melanie mentioned, we've been funded by the ODA Specialty Crop Block Grant, the Northern Nut Growers Association, the Policy and End-to-H Warner Endowment Fund, and CFAES Seeds. And with that, I will take questions.
So we'll go 5 minutes and take
Yeah, about
questions.
5 minutes for questions. Any folks here before we? Yeah.
Really?
Did you check if the Caulototricum can infect chestnut roots?
I didn't inoculate any roots, but I did isolate root tissues, and we never found it growing in the roots.
Interesting, because a lot of Caulototricum will just infect the whole plant.
Yeah, and that's not to say that it couldn't. It's just the roots we looked at weren't infected. So,
Okay.
you know, we didn't do an extensive root survey, and that's something I'm curious about as well. But what we did look at, and in fact, some of the twigs, the seedlings I inoculated for the pathogenicity tests, after they were showing symptoms, I took some root samples and plated root samples and didn't see any in the roots. So I don't think it's a common thing in the roots. Yeah, Chris.
As it relates to the infection within the seed, is there a toxic front that you see that is before the hyphae gain entry into those tissues? Is the material in front of it dead, indicating that maybe there's some sort of secretable toxin or anything like that?
Yeah, that's a good question. So so the question is do we see an infection front? Do we see necrotic tissue kind of ahead of the infection in the kernel? So if that was the case, so there's the vascular tissue that connects the kernel to the twig, basically there's kind of they call it the funiculus, the little tube that connects the vascular tissue of the tree to the nut and the placenta at the top of the nut, through which the nut is sort of filled and fed sugars. At harvest time, that funiculus, those vascular tissues have already senesced. So at harvest time, we really can't see, it's not obvious what causes, like, you know, there's already dead tissue anyway. So you would have to look at those tissues kind of throughout the development. It'd be nice to know if you knew you were going to have a heavy infection year, and you knew you're going to have a lot of disease, to be able to analyze the funiculus tissue and see if it's actually dead ahead of the spread into the kernel. The way, for what we were doing, we had no way of seeing that. But yeah, that would be that's kind of like, in my mind, that's like the holy grail, right? You got to see, you know, how is that infection occurring in the kernel? If it's happening through the funiculus, it would be interesting to see that sort of dying ahead of the wave.
I was thinking just even in your stored kernels, specifically when you have them at like at 28 degrees, you have the material is moved through that stored material. So if you were able to look at that stored material as it's progressing,
Mm.
you know, do you see a toxic front as it's moving through the endosperm type of tissues?
Yeah. It's hard in those warmer temperatures because those secondary spoilage fungi pretty much overwhelm that original anthracnose infection. So it becomes really difficult to see like that, whether the necrosis is caused by the secondary spoilage fungi or the anthracnose. Yeah, so even within the kernel, it seems like once they're off the tree, and all those other fungi take over, it's just really messy. Who knows what's going on at that point? I think if we had, you know, more advanced techniques like qPCR or something that was specific to Cholestaticum henninense, so we could look at abundance of fungi in specific tissue, not just presence absence, that could be really helpful. So I think having a Cholestaticum henninense specific primers for qPCR would allow us to really see these hotspots of infection in different tissues over time. Anybody else here?
Have you abandoned the idea of investigating a relationship between the infection and pest, like the wasp, or since with the Cox postulate you saw that you had to wound the tissue, and then you pass to the hypothesis, okay, living endophytically, but are you also thinking to focus on other hypotheses or just on?
Yeah, so the question was, what about the relationship between pests and disease? So because we saw, wasp infections correlated with disease. So I guess the short answer to that is, from what we've seen, I don't think there's a direct relationship between pests and disease. Like I don't think either the gall wasp or chestnut weevil or periodical cicadas or these insects we see in the orchard are spreading the disease themselves. But I do think the stress on the trees caused by the wounds from these insects could make the trees more susceptible to disease. So I do think there is an indirect relationship between pests and disease. And it might be interesting to do something like, have an inoculated tree and just poke it full of holes, kind of simulate insect wounding and see if we see more disease development under those conditions than without. How about anybody in Columbus or anybody on Zoom? Any questions from the Zoom audience?
You have a few questions.
Okay. Can you read them? I can't.
Okay. I don't know if you can hear me. The first one says, do you think eliminating very sensitive trees from the orchard might reduce the disease pressure on more resistant trees?
Yeah, so if we eliminate more susceptible trees, will we see better disease management overall? That's not a bad strategy. I think, most growers would kind of default to that, right? You would say like, okay, well, let's just get rid of all the highly diseased trees. I can't say confidently whether I think that would make a difference to the trees that remain. The fact that we see trees with high disease incidence and low disease incidence more or less side by side makes me think that infection in one tree is not necessarily causing infection or leading to infection in another tree. So I think these infections are somewhat independent of each other. So I don't think culling the more susceptible trees would make a difference in the overall disease pressure, but it just might make your harvest easier as a grower. So you're not messing with the more diseased nuts. Yeah, one more or?
Yeah, one more.
Yeah.
Okay, Celia says, I don't know a lot about chestnuts, and there's a few questions here. So, first, what is the cultural distribution among growers? The second, which is more prevalent than queen? And is there any difference in customer preference? And the third one says, why people still plant peach if it seems to be much more susceptible?
Yeah. So questions about the cultivar, especially the peach that's more susceptible and Ching that's more resistant. Both of those cultivars are planted widely because the nut quality, other than this anthracnose disease, the nut quality is really good. So the trees are really productive. They produce reliably, they produce relatively big crops year to year, and the nut flavor is good. The nut size is preferred by consumers. So these have been selected for their nut traits. And it's only just recently we've been seeing this difference in anthracnose disease. So I think at the moment, nobody has planted a chestnut orchard thinking about anthracnose susceptibility. They're all thinking about sort of the characteristics of the finished nuts. And so maybe in the future, growers that are planting new cultivars might start considering that. But at the moment, growers are just planting based on those qualities of the nuts themselves from the consumer standpoint, and they're both really good nuts. Peach is named peach because the shell is actually kind of fuzzy. It's really distinctively fuzzy, so that's why it's called peach. So anyway, they're both very charismatic cultivars. So it'll be hard to fully get rid of the peach.
One final question. But go ahead.
I would just like to offer a reverse acknowledgement that Route 9 Cooperative and the chestnut growers at large appreciate the work that you have done and that Ohio State has undertaken, because this is a really important problem for us, and we didn't have anywhere to turn to get answers. And so this is hundreds of thousands of dollars to cost to growers to deal with this. So this is really important, and we appreciate what you have done, even if you haven't solved the problem. Just knowing about what the options might be, and don't bother spraying, things like this are important. Thank you.
Right, that will end.
Thanks everybody who joined us on Zoom, wherever you may be in the world. Appreciate your interest, and hopefully many of you family friends hopefully talk to you sometime soon. Enrico and Mitch, we will switch over to the other Zoom link here shortly.
You probably want to try next 10 minutes away.
Yeah.
Oh,
Oh, yeah. I also forgot to read out the